Hello folks, my name's Ahad. I'm a software engineer here at A&D. I used to work directly with OpenScope, and now I work on a team of scientific computing and kind of support all around. And I'm going to give you guys a little bit of a introduction to our NWB conversion pipeline, what NWB is, how to use it, and the kind of general tools that are good with it. So first of all, it sounds like Ariel and others have already kind of discussed pipelines, but I'll still go over them just a little bit. So all of our pipelines exist on a platform called CodeOcean. It's a reproducible and traceable computational science platform. And what that means is that we can generate these pipelines and capsules, which are able to be shared and reproduced by anyone. They contain the code, but also Docker images. So if somebody has a capsule and they share it online, they get your exact environment, code, data, etc. Also, you can take the pipelines that we create and run them locally if you need, though CodeOcean is a cloud platform for a reason. And again, like Dan was going over in detail, we have a standardized schema which allows these platforms to be pretty plug and play for A&D data. If it's following our JSON formats, it should be able to read that. So why do we need our pipeline specifically for NWB conversion? Why not just share the raw data or all the individual files that we processed? Well, first of all, each session has hundreds of gigabytes of data, and these are in tons of different formats, NumPy files, CSVs, H5s, pickle files. And it's not really easy to share that much data in that many forms. My understanding is in the past, the people used to actually ship hard drives and then download them onto their local computers. And that's just not convenient at scale at all. Also, many hands are touching data, which means like someone may be in charge of pre-processing, someone may be in charge of QC, someone may be in charge of packaging, and having that many different files handled by that many people is just a recipe for disaster if you aren't standardizing how it's done. So creating pipelines that everybody can use in a standardized way helps prevent a lot of confusion that way. And not only are we kind of standardizing the code that we're using, we're standardizing the data in and out. That means that we can use a capsule that will handle any CamStim specific project and generate like a STIM table for it, for example, or something that can handle motion correction for all the open sessions here in EC2. And out means that we're generating NWB files on dead. We don't have to share intermediary files. We don't have to share an Excel sheet that says this file is here and this file is here. We can just upload a dandy set and tell people this is this mouse with this information. And also it just helps kind of facilitate communication a little bit. If I want to ask somebody what they're doing on code, I can just send them the pipeline and show them, hey, this is what we're doing. Have you guys developed a similar tool? Or can you run this with your data and confirm that it works? And similar with external teams, they just get access to CodeOcean and do those kinds of things. Now, I've talked about the pipelines and the kind of standardized data, but you may not actually know what an NWB is. And maybe it's like really confusing if people have been talking about NWBs all day and you're just not familiar with them. So I have a tendency to call them NWB files, but it's actually not just the files. It is a standard ecosystem, which means there are kind of data formatting and schema and all sorts of other things that are standardized and open source. That means that for eFIS, OFIS, different stimuli, modalities, all this kind of stuff has been described in a way of it should go in this format, it's going to be done this way. And also there are resources available that aren't just related to the files that Carter's going to go into detail a little bit more later. And this whole effort is created and supported by the Copley Foundation and the NIH Brain Initiative. And because of that, that means that it's really well documented and supported. So you can always go to their libraries and just check. And they have really active Slack channels and stuff that you can ask for help with. And then going over some of the standardization that I described earlier, the schema is really standard but flexible. That means that, for example, there's an acquisition section, an analysis section, general, intervals, processing, etc. And each of those are kind of intended to be used to store a specific type of data. Like acquisition should be storing experimental raw acquisition data, while analysis is going to store information about analysis and figures. Processing will store the data from acquisition that's been processed, and so on and so forth. And then you can organize your NWBs in standardized ways because of that. And tons of stuff is supported by this tool called NWB widget, which will allow you to actually, as long as you're following these standardized formats, visualize your data and see it and share it. You can create a table and look at all your stimuli and your intervals, for example, or a picture of all your running information. Now, a little bit of a warning is this is really still flexible. So in theory, you could put data in the wrong place or, put data in kind of a weird format or something. So I would really suggest that you read the schema and make sure that you're doing things correctly and not just doing what immediately seems correct from the schema. So if you have your NW files, you understand the format, maybe you generated them with our pipeline or you generated them yourself, you may be wondering, well, how can I share them? And Dandy is a really good platform for this. It's able to publish and process some neurophysiology data. It is quite large. Last time I looked at it, it was 500 terabytes with 300 plus Dandy sets. I'm pretty sure they're like 600-700 now, probably more. So it's really big. And you can share files and your Dandy set as easily as sharing a link, and it will generate a DOI. And essentially, instead of data sets, they have these things called Dandy sets, which are just storage of an experiment or project that has all the mice associated with it. And you can embargo it if you have a grant or something to embargo it, or you can open it to the public, and you can make drafts before you do release, so you can adjust layouts and stuff. And whenever we do a data handoff or release, we will do like a draft embargoed, and then eventually click the button and have a nicely formatted Dandy set for everyone to look at. And then in addition to storing your files, there's this thing called Dandy Hub on Dandy, which is this kind of powerful processing environment that allows you to do your analysis or process or some post-processing if you want or anything like that, all on the cloud, directly connected. So I've kind of discussed how files are generated and how they go up on Dandy, but there is information that's not necessarily in the NWB file that's helpful to know. So some of this metadata we stored specifically for OpenScope on a website called the OpenScope Data Portal. So you can see, for example, in a really easy format, which mice all belong to one genotype. You don't have to download all the information. And this is filterable in that kind of way, so you could find all the mice that all the sessions are associated with a specific mouse ID, or all the mice that are a specific age, or things like that. We also share background information on the project, so you can see like the experimental design descriptions there. And initially, this is kind of hidden for a year with the teams where we kind of develop their Dandy set and it's embargoed. And then once we click the release button, this becomes available, and we add that specific Dandy set to this publicly released section, and you're able to click on it and download files from Dandy. And if anybody wants to scan the QR code, you can go there, but it's not necessary. And yeah, that about covers my section. I've talked about getting files and how we share them, and Carter's going to talk about some of the standardized analysis tools, what you can do with our data.
Okay, thanks, Ahad. Okay, so my name's Carter. I work on the team with Jérôme. I'm an analyst for OpenScope. And my main project for the last 3 years I've been here has been the OpenScope Databook. So as you saw yesterday, we have a ton of infrastructure for gathering really high quality data. We have a ton of expertise. And we also have a ton of many platforms and a lot of online resources that we've been using to package our data and process our data in a really robust way. So because we put all that effort into making our data highly usable, highly accessible, we wanted to kind of, punctuate our whole processing pipeline with a way to actually disseminate the information and assist people in actually using it. So the state of affairs 3 years ago was that we had our data in NWB thanks to Ahad. But NWB's, the ecosystem was still kind of immature at the time, not super familiar to a lot of people. And there's also no central repository for like analysis code that's super, super common, right? Which I'll go through in a little bit, some like really common analytical techniques. So The goals we set out to achieve with the Databook were we wanted to create this one centralized place. We wanted to be able to perform a lot of common analyses on our data with just the click of a button or two, make it really easy. We wanted to provide the code to access our data off of Dandy. And importantly, I mean, you guys have gotten, you know, the hands-on, up-close look at all of our stuff, but most people don't have that fortune, right? So it's really hard to understand all the work that goes into our data and how to use it and what a lot of it means, unless you have it documented really thoroughly, right? So we wanted to educate the users on our data with the Databook, and then obviously generate the figures and share the figures. Ultimately, I mean, the goal of OpenScope is to help, you know, reproducibility, usability, open access to our data. So that's what we wanted to do here with the analysis. So the way we did it, we settled on a web-based platform with Jupyter Book. So Jupyter Notebooks was the obvious solution here, right? Because you can interweave code and documentation. So Jupyter Book is essentially, that's just a website that's just a library of Jupyter Notebooks. And in our case, it's hosted for free on GitHub Pages. And importantly, it doesn't just have access to the notebook document, which is, you know, just the static code and markdown, but it also has, we integrated several launchable computing platforms that you guys can use freely. So instead of having to put all the work of setting up the environment, hopefully a lot of that is done for you. And it's linked to the GitHub repository also. So this is not exhaustive, but this is a good overview of the kind of stuff that we have in the Databook. You guys have seen our ePhys and our optophysiology, right? So the Databook primarily focuses on our neuropixels data and our 2-photon data, and doing all the analysis directly from the NWB files. It has a lot of visualizations. Important stuff that it might have that you guys might be interested in is like opto-tagging or generating receptive fields. These are super common, and there wasn't any centralized repo to just do this automatically for you. So we can do that now from our data on Dandy. And then there's also some higher order analyses like GLMs, tensor composition analysis, and CBRA. So we'd really encourage you to check this out if you're trying to launch into our data really quickly, right? You want a good starting point. So I mentioned our computation a second ago. So we put a lot of work into trying to make the analysis really easy to reproduce, specifically to reproduce the environment. Many of you might know that it's really hard to use someone else's repository and keep their environment working in Python. So we did all that we could to make that happen really easily and quickly. Ahad already mentioned that Dandy Hub, which is what I would recommend for the main platform here. Dandy Hub basically gives you the free compute as long as you're not using it for any nefarious purposes, and it allows you to set up the environment with Docker. And another good thing about Dandy Hub is that it has direct server access to the data, so it's really quick to download the files and or stream the files. If you're more familiar with Google Colab, we have that as well. And of course, you can also just download an individual notebook and run that, or you could clone the GitHub repository and use it that way. Let's see. And then you might have heard a little bit already about CodeOcean. So we've integrated a lot of the on-institute's processing and packaging into CodeOcean. So we did that with the Databook as well. And it works, yeah, it works well. And soon this will be publicly available for other people as well to use. Let's see. Yeah, it also has modular capsules and things like that. So I'll demonstrate this in a little bit. To access the data, Ahad already mentioned a little bit, it's all on Dandy now. So you can essentially just download the files through the Dandy GUI, or you can use the Python API to stream or download. And embargo Dandy sets also you can access with an API key. So might as well just show you everything we've been talking about, because that'll be much quicker and clearer. So if you're interested, I would start by just going to Google and looking up OpenScope Databook. Don't just look up Databook, that's something different. And you can see the GitHub here. Let's see. So we have the GitHub repo. Some things I want to point out is that we have like a citable resource here at Zenodo. So we wanted to treat the Databook like a fully fledged publication. So it's citable here. It uses citations in the document, and it has an authors list. Let's see have a table of contents. So let's actually go to the website here. So this is the Databook. This is the authors list also. Because part of OpenScope's mission, right, is we want to promote open science. The Databook is also open for contribution. And I think Dan mentioned earlier, like, ideally, going forward, a lot of you guys will help contribute to the ecosystem, contribute your analysis and things like that. So one thing about the Databook is anybody who makes a PR and undergoes our review system, gets integrated into the main branch, they automatically get added to this authors list, which is pretty cool. And we have different views of how different contributions happened. Here on the front page, we have an explanation of how to use it and more in depth. Okay, and then I want to go over a few of our exemplary notebooks, just to show you what kind of stuff we have. So let's look at this one here. So this one just really quickly shows you how to access a file. If, for instance, you want to look at OpenScope's Vision to Hippocampus project, you just go here on Dandy, you can go to a file, and you can click this I here and get the file path. And then this is the Dandy set ID. So you copy both of those in here, and it will download the file for you. Alternatively, you can stream it, and you can access the file and open it really quickly with this code here. So you can see everything we have in here, which is explained in more detail throughout this chapter. And one example I'll show here is this one. So LFP is common in electrophysiology. So this is how we access LFP. We get event-aligned responses in our LFP data this is our visual coding data set. And so you can go down here, you can use this notebook, type in, the data. Tiny things might vary between data sets, like this path here, but for the most part, it should be pretty easy to just replace it, and then it will access all the data properly. For like this, with the name of this stimulus will be different, for instance. But you can see that up there is accessing the LFP. This down here is accessing the stimulus table, right? It's just a table of all of the stimulus events that occurred throughout the session. In this case, it's just selecting when the orientation was 150 in our stimulus for the gradings, but you could easily change this to suit your own purposes. And then it will interpolate it for you. It will get these time-aligned windows, and then here you have this really nice, clean LFP, like event-aligned responses here, right? And so this is along depth, so this is superficial, and this is deep. And ideally, you could plug any of our Allen Institute data sets into this, and it should work with very minimal changes. And we can do even cooler stuff, like get, receptive fields. So with our 2-photon data, you can do essentially the same thing, but here we'll calculate the receptive fields for one of our data sets, which is really nice. We have our higher order analysis here that I mentioned. I would encourage you guys to check that out. And I want to draw more attention here to this chapter. So for each of OpenScope's data sets that we've collected over the last 3 years, we've made a notebook. We collaborated with the external team to make a notebook that specifically explains their data set and their experiment. So if you're interested in any one of these, I would look at it because it's a really good starting point. Let's see. Like this one, for instance, the team wrote this, it explains the experimental design. Actually, I'm going to go onto CodeOcean now and show you guys there. Let's see. So this is CodeOcean. Soon you guys will be able to access it publicly, but we have a public collection here. You would just go to codeocean.allenneuraldynamics.org. I think you have to make an account, but you'll be able to see our capsule here and launch it. This is what it looks like. And then you can launch a workstation. In my case, I did VS Code. And it allows you to run the notebook. So this is our VIPO dataset, which we released a few months ago. It explains here, it's basically, a lot of it is just a vertical moving bar and some natural movies. This gives you an overview of the dataset. Basically, these are all the files in the dataset. We pick the file to download. This shows you the probe track layout. these are images that are saved in the Databook, but they were generated procedurally from what was in the file, from the coordinate information. And you can see warping, that's an artifact from registering the real mouse brain to a model mouse brain. The probes don't actually bend quite like that. We extract our unit spikes information here. It shows you how to do all of that. Get the electrode information. You can sub-select them based on brain region. Another thing we show here is the session timeline. So these are the names of the stimulus, which the actual content of those stimulus can be viewed elsewhere in the file. But this basically shows you what was shown throughout the duration of the session. Like we already did, you can extract particular stimulus, the timings of a particular stimulus events you're interested in, and it will get a time-aligned response matrix here and plot them. These are just random, so you don't see really clear responses, but you could sub-select particular units based on their region or the noise level of the unit, basically the quality, and then also additionally you can get something like the receptive fields here, which once again they're not very good because these units weren't selected very clearly. But yeah, so you could, like I said, you could plug this in with any one of our data sets and it should, for the most part, work. So I would encourage you guys to look at the Databook and look at all of this code here. Okay. All right, a few more notes. Okay, a few more notes is the key challenges. The environment is obviously a big challenge. That's really hard to replicate in Python. NWB files vary, like I already mentioned, so that's kind of an intractable problem long term. But for the most part, it's very streamlined. Outlook. So, like we talked about, we want to encourage people to contribute, and we want the Databook to become more expansive and include other people's analyses as well. So, as people develop analyses on their own data sets, put them on Dandy with this sort of standard file structure, we want people to basically help disseminate their own findings to the Databook as well. And we also leverage GitHub, basically GitHub PRs to have a rigorous review process so that things are actually reviewed by external scientists. So we know that the plots in there are clear, they're clear findings, and like I said, we'll accept contributions from many people. So, and actually tomorrow we're accepting a notebook from the Flatiron Institute, so it's always growing. Yeah, I just wanted to acknowledge many people at the Allen Institute and outside the Allen Institute. This list is not complete. Too many people to fit on one slide, unfortunately. Thanks. Questions? Yeah.
So just to understand the general structure and how it works regarding the publication, the data sets. So for example, every experiment on the OpenScope platform will be published here, or some will not?
Yeah, so OpenScope specifically has a commitment to publish our data publicly on Dandy after a year.
Okay.
So
After a year. Okay.
yeah, so it's private access for the team for a year, and then after that, it's publicly available. And we'll continue to add notebooks for our released Dandy sets to help analyze it.
And if there are, you download, for example, a data set, and for some reason it is revised, there are some corrections to the metadata, I don't know, updates, do you flag it somehow? Do you notify?
That's a good question. We have done that before. We haven't come up with any particular way of doing that, but that's a good idea. We could note it on the Dandy set. But yeah, that does happen sometimes when we do update the data.
Yeah. And if, I'm sorry.
Whenever you release a data set on Dandy, you get a unique DOI.
Okay.
So the data set will get a new DOI. So
it's a way to say something happened.
I think you keep access to all, like it's permanent. Versions of that's my understanding.
One more question. I'll remember later.
Yeah, of course, you can find me. Sorry. Anything else? Great, thanks.